Hello everyone, I'm Rike. I'm a PhD student at Cubic. Gisela introduced Cubic briefly this morning. It's a bioinformatics core facility, and we are fairly involved with the NF-core community and contribute a lot. And this is also how I got involved in the NF-core sarek project, which is a pipeline for somatic and germline variant calling. And it's named after a Swedish national park that you can see here on the picture. The pipeline's been around for quite some time. It was first started in 2016 at the Karolinska Institute, NGI and NBIS, and then ported to NF-core in 2019 and published. And then sometime after this, everything changed and DSL2 came out, and we rewrote the whole pipeline and then pushed the first release earlier this year. And all release names are based on landmarks in the Sarek National Park, and since I don't speak Swedish, I will stick to just using the version numbers. So there's a pretty fluent developer base for the pipeline. I think currently probably we have like 11 or so relatively active code contributors from all these various institutions you see here. But since the pipeline's been around for so long, there have been lots of contributors, lots of people who have made feature requests and found issues and helped us make the pipeline better. Before I go into deep, just a short slide on the background. So for variant calling, oftentimes you have like a tumor in a normal sample, you extract the DNA, sequence it, and then align the FASTQ files of each of these samples to the reference genome, do some cleanup, and then in the end try to find the difference between your sample and the reference genome, but also between the samples to find tumor-specific variations. The pipeline can handle various input data sets, whole genome, whole exome, and panel data. It was originally designed for human and mouse, but can be used with other organisms as long as reference files for it are around, and it's for short reads only. So a brief overview of the pipeline. We've also made one of these subway maps that are now around everywhere, I think in NF-core. So the pipeline starts with a big block of preprocessing, followed by variant calling and then annotation. And here you can see we have round blocks and squared blocks, and only the squared steps are mandatory to run. And the round blocks can be turned off or on depending on what you need. To go into a little more detail, for the pre-processing, it's based on what's recommended by the JDK best practices. So first, the reads are aligned to a reference, and we have currently 3 different aligners available, then duplicate marking, and then base quality score calibration. And then this is followed by variant calling. We have 3 different tracks, so germline samples, the normal ones in blue, then tumor-only in red, and paired variant calling in blue and red here. And there are various tools available for each of SNPs in the structure variants, copy number callers, or microsatellites. And also all these tools can be run in any combination that is useful for the analysis. And then the last big block is the annotation, so assigning information to variants, such as predicting effects on the protein structure from a variant, or also annotating it with information found in public databases like Linva or Cosmic. And at the moment, we have SNPF and VEP for annotation available. So then, at the very end, after each of these steps of the big ones, we are running QC tools, and then all of these are collected in a multi-QC report and published with the results to check that everything worked and the samples and qualities as expected. So for the push to DSL2, of course, I just wanted to highlight some of the major features. So of course we ported it all to DSL2, and I'm quite happy to say that pretty much all the modules we use in Sarek are now part of NF-core modules, or we've also benefited a lot from all the modules that were available already when we started the conversion. And also like now when somebody updates, we have a version update in GDK or something, we just pull the modules and it all works very nicely, and it's a lot less overhead for us. Then we added PyTest on pipeline level, so Harsh will briefly introduce this for modules earlier, and we figured we can do the same thing for pipeline level, so Edmund and Anders have added this, and we check MD5 sums for all the tests that we have. on each pull request to dev to make sure we don't break anything, or if we do, we are well aware of it. One aspect of this release was also focusing a little bit on optimization. For this, one of the points with Sarek is always that the results file can be quite big. So after duplicate marking, we are now compressing everything to cram format and then run the rest of the pipeline on this, reducing the results file size. And also, while Nextflow handles, of course, implicitly the parallelization for all the samples, for each sample, we also split it more, run mapping, for example, in parallel over multiple charts of FASTQ files, or parallelize across intervals for base quality recalibration and variant callers. And all of these features can be tuned by parameters to determine how much splitting you want to do, or if you don't want to do any at all. And maybe to highlight this briefly, we benchmarked this here on two smallish samples. So what we can see is basically on the more splitting we do, the faster we are, but we do pay this in storage space. And we've also seen, similar to the talk this morning, that when we run things on AWS, it might sometimes be more beneficial to not split up as much. Right, with this, I'm also almost at the end of my talk. I just want to give a short outlook. When we ported everything to DSL2, we couldn't really port all the features that we wanted, or we would still not have released, I think, because the pipeline is quite big. But we are continuously working on adding back in these missing features. And one of them is the send-high-end support that multiple people are currently working on. Yeah, we, of course, continue improving it and adding new tools. I think just this week we are possibly getting 2 more aligners in with the new sub-workflows, and also porting everything now to our sub-workflows to NF-core, but also taking in the NF-core sub-workflows that there are into Sarek. Yeah, and we have started some benchmarking. I just showed you a very short slide on this, and we are repeating this now with bigger and more samples, and we'll publish this. So, yeah. Keep an eye out for that. Yeah, and then last but not least, this was a huge effort of a lot of people in the NF-core community. Maxime, of course, as the father of Sarek, so to say, then Anders and Edmund added a lot of the testing. Lasse is doing a lot of benchmarking. Gisela also added like AWS full-size tests for us now. We have Susanne and Oscar doing copy number callers, Francesco, Nick, and Gavin doing GDK and FG Bio sub-workflows. So it's really been a big community effort to get it to where it is today. Yeah, and last but not least, we are everywhere. We're of course on Slack, GitHub, the website. If you whisper Sarek, one of us is probably going to show up. Get in touch, join us in the development, or if you have questions for usage, we're here.
So I have a question from Aaron. So he's asking, which variant callers have performed the best? Are there any you wouldn't recommend?
That's a hot topic. So we've done some benchmarking internally for germline variant calling, and there Friederike has performed the best for us. Also deep variant has been performing really well, but I mean, they've all been performing really, really well. It's like minuscule differences between them.
Okay, thank you.
Thank you.